Let me share this. Yeah. So today we are just giving ourselves introduction to what we are. So by next week, it's going to be a proper add-on practical. You understand? But I'm just trying to lay a good foundation, a solid foundation in our mind to know what we are about to do. Now, artificial intelligence in medicine, and we are talking about computer-aided drug design. CADD, computer-aided drug design. First thing, what is a drug? I know even though I know more than 90% of people on this class in this class are pharmacy students or science students. But I don't know, we may see have some that are not called into a science, that are not called science students. And that's why, so when I ask the P2, P3 pharmacy students what is drug, don't say what is Dr. Who asking us. I know what I'm saying. You understand? So a drug is a medicine or any substance which has a physiological effect when ingested or otherwise introduced to the body. Yeah. So any substance. So to some extent, you can refer to water, even the water that you are drinking as a drug. So whatsoever that you take into your system, either through the mouth, injection, or even the body cream that you rub, you understand, can be referred to as drug because in as much as they can bring about a physiological effect in your system, in your body, you can refer to them as drug. So when we're talking about drug, it's not necessary. We are not only talking about those ones that you go to Walmart to buy. You understand? No. In as much, even when you are thirsty and you are hungry, and you go and get a burger, water burger or anything, the moment you take them, what happens to your system? You feel relieved, right? So that they have had some physiological effects in your body. So they can be referred to as drug. So anything that you take, any substance, that's why I say a medicine or other substance, which has physiological effect when you ingest or otherwise introduce to your body can be referred to as drug. All right. So what is drug design? Drug design is the incentive process of finding new medication based on the knowledge of a biological target. So in this place, as I'm giving the definition, I'll be explaining some key points. Now, an inventive, when you say something is inventive, that's something that is new, novel, research process of finding new drug based on the knowledge of a biological target. You will be asking, what is a biological target? Thank you. When when you are designing a drug, you understand? The first thing that should come to your mind is, this drug that I'm designing, what disease is it going to cure? Or have you ever seen anybody that says, I've designed a drug, and when they ask him, this drug that you designed, what disease is it going to cure? And he's saying, I don't know. Have anybody ever experienced that before? Am I making sense? So can you just imagine you saying, yes, as a pharmacy student or as a pharmacist, I have a drug now. And they ask you, what is that drug going to do? And you said, I don't know. Is it going to cure headache? Is it going to cure? So what I'm saying is, every drug in market has a target they are focusing, right? So whether your drug is curing HIV, SARS-CoV-2, is curing cancer or anything, there must be a target that your drug is focusing on. So the process of finding a new drug based on knowledge of a target is referred to as drug design. When they produce Tylenol, right? Tylenol is produced to cure pains, to cure fever and headache, isn't it? So because people have the experience of headache and the rest, okay, what causes headache? Then they say, okay, there are some enzymes, there are some mechanistic pathway in the system that causes headache. So let's design a drug that can alter that pathway so that there will be a cure to headache or fever. That is when Tylenol was being produced, right? Now, when they produce drugs like lopinavir is an antiviral drug, right? Anti-HIV drug. They didn't just go from somewhere and just bring lopinavir. No. Because of their understanding of HIV virus, they were able to produce a drug. So when you are designing a drug, you have to have an understanding, you understand? Understanding of the disease that you are preparing your drug against. Am I making sense, please?
Yes, sir.
Yeah. Yes.
So nobody just sit down and design a drug without a particular disease. No. It doesn't work that way. So even though your drug, you can design a drug, and the drug that you're designing work for another disease, apart from the one you have in mind. But the first thing to drug design is to design based on the knowledge of a target, of a disease. So that process is called drug design. So I think it involves the design of molecules. So the molecules may be chemical compound that are complementary in shape and charges to the biological targets with which they interact and therefore bind to. Let me explain this. I know we have a lot of pharmacy students here, and there are people that are not pharmacists. So when I'm throwing this question to you, everybody, and I want a response. When you have headache and you go to the pharmacy and say, please, I have headache or you have fever, and they give you ibuprofen, and you take the ibuprofen, and after some minutes, maybe like 5-10 minutes, that headache or fever stop. What do you think the ibuprofen has done in your system? I'm throwing this to everybody. Let's interact.
Okay, I'm guessing the pharmacy student should probably start talking. So I'm a pharmacy student. So
you are a pharmacy student, right?
Yes, sir.
Let's start with
you. From what we understand, most times when we feel pain is due to inflammation.
Okay.
And the NSAID you mentioned, what it does is that it is an anti-inflammatory.
Mm-hmm.
And what it does is that it goes to the causes of inflammation and stops them. So okay. I'm guessing that's what the medication does, But of course, if I have a headache, I don't care whether it does inflammation or not, so long as the pain stops.
Yeah. Thank you very much for that response. Any other opinion?
I think it will be really hit what the main thing does. It usually has like a target site where it has a main target site where it would affect go in the body to heal whatever illness or symptoms the patient is feeling.
Yeah, thank you very much, Timilan and Obina. That's a very fantastic response. Yeah. So when you take your ibuprofen to the system and you just discover that the headache or the pain stop, you understand? The ibuprofen has not gone to your system to do magic. No. What ibuprofen has gone to do is to go, I think, as far as headache is concerned, it has to do with prostaglandin, right? Prostaglandin are special set of hormones in the system. So when you take your drugs go to where prostaglandin is being produced, right? Bind to the receptors on the production of prostaglandin and reduce the production of prostaglandin in the system. So that site where prostaglandin is being produced, the structure of your drug must be complementary to the binding of that drug, to the binding of that site of production. Another example, physical example, when I have a key, right? And let's say I go to Obina's house, isn't it? And I have my key, and when I get to Obina's house, I observe that the door is locked. And I take out my own key, try to insert and open, and it's not opening because the shape of my key is not complementary to the shape of the lock in Obina's door, right? So the same way, that's why the drug for headache cannot cure HIV, because the shape of ibuprofen is not complementary to the shape of HIV viruses, target proteins. Am I making sense? So that's the way it is. So your drug must have a complementary shape and charge to the biological target responsible for that disease that you are curing. And that is the simple truth of it. So the shape of your drug must be complementary to the biological target that you are looking at. So if you want to cure SARS-CoV-2 spike protein, go and look at the drug. The drug that you are going to use to cure SARS-CoV-2 spike, to stop SARS-CoV-2 spike protein, must be complementary in shape. Just like your key and your lock must have complementary shape before they can open each other. So they must have complementary shape and charge before they can work and cure the disease. And that is what our drugs, most of the drugs that we are using, that's what they do. Now, drug design now. So the molecule that we are talking about will interact with the target, right? Bind to the target. When they bind, they either activate or inhibit the function of that target, such as protein. Do you understand what I'm saying? Let me give you another example. We are going to go more into science. When people are talking about Alzheimer's disease, studies have shown that when Alzheimer's disease, when acetylcholine esterase leads to enzyme, catalyze reactions, and produce more choline in the brain, it can result to Alzheimer's disease, right? So what people are now doing now is, okay, can we look for a drug that will be complementary in shape to the acetylcholine esterase enzyme, thereby stopping the production of choline? So in that case, they are looking for a drug that can inhibit the activity, of acetylcholine esterase. So when you inhibit the activity of that enzyme, it will stop the production of choline. That is it. So in this case, the biological drug must inhibit. Inhibit means stop or reduce the function of the biological molecule, in this case, acetylcholine esterase, from producing choline. Because when choline is produced, there's going to be Alzheimer's development. Do you understand my point? That's that drug. There are some other drugs that their own is to make sure that the enzyme is functioning well before the disease can be produced, right? Let me give another example of diabetes, right? There are insulin-dependent diabetics, right? So insulin-dependent in the sense that when you take your food supplements, when you eat your hamburger, you take your tea, or you take your coffee or anything, there are carbohydrates in those foods. Am I correct? When you take them, it is the role of the body to break those carbohydrates into small units called glucose. Am I correct? When you put them into small glucose, it is the work of the insulin hormone to clear excess glucose in your blood and convert them to glycogen. Now, for someone that is suffering from insulin-dependent diabetes, the insulin is not functioning enough, or it's not being produced enough to convert excess glucose in the bloodstream to glycogen. So in that case, it's either they ask the person to go and be taking a drug that will activate insulin to clear the glucose in the system, or they will ask some of them to start injecting themselves with insulin hormone. So what I'm saying there is, whatever drug that you are designing is either inhibiting or activating the function of a biological protein, a biological molecule, either a protein, enzyme like acetylcholine esterase, or hormone like insulin in the diabetes example I just gave to you now. Am I making sense, please? Are you getting my point?
Yes, sir.
Yes, sir.
I'm just trying to take time to lay this foundation. So by the time we start doing our software, we start doing our computational stuff, and you see us bringing this protein to this drug and the rest, you will understand what you are trying to do. That is why I'm taking this time to lay this foundation. Okay. Now, this molecule that we are talking about can be organic small molecules. So drugs can be a chemical compound. And I know we might have seen a lot of drugs, right? In different color, in different shape, right? So they might be organic small molecule, complementary in shape to the target. Yes. That is why the drug that you use for headache cannot be used for stomach pain, because the target for headache is different from the target for stomach pain. And when there is a difference in targets, there's going to be difference in the shape of the target. And there's difference in the shape of the target, then the drug that is required for each of them will require different shape. That is why a single drug cannot cure all disease. No. Because every target for every disease has their specific shape of the drug that is required. So the drug molecule must be complementary in shape and in charge to the drug that you are using. And by the time we start our real practical next week, you will understand what I'm saying. So now, we're not talking about drug design. I'm telling you, this diagram that you are seeing, that's what it takes to design drug. And next week, by the time we start our work, everybody will have to hold their laptops, right? Even though I will advise that you can have a laptop, but still look at what I'm doing. Then later, and that's why if you have meeting on Friday, before the next Friday, take time and dedicate at least 2 hours to whatever practicals that we've done, so that you can replicate, you can do the same thing on your own. Now, drug design. Drug design has to do with, don't forget one thing, when you are trying to design a drug, the first thing you have to understand is, what biological target am I focusing? Am I targeting? Because it is that biological target that you are focusing that will determine the shape of the drug that you are designing. You know, just like people that are into house building, before they start designing the home, they must have an idea of what is the size of the land, the square feet of the land that is available to build, right? So you can't say you want to build a 5,000 square feet house on a 2,000 square feet land. Is it possible? So it's not. So the same way, when you are designing a drug, you have to have an understanding of the biological targets. So let's use SARS-CoV-2 for instance. If you want to, for instance, if you want to cure SARS-CoV-2, right? You can look at, okay, how can I stop SARS-CoV-2 from getting access into the human body, right? So you can look at, okay, the first thing you do as a scientist is, how do SARS-CoV-2 gain access to the human body? And from research, it has been shown that when SARS-CoV-2 virus get access to either the eye, the mouth, or the nose, that is when it can gain access to the body, to the body. It's not through the skin. It's not through any other means. Even it's not even, it's not even through sexual intercourse, right? No. But basically, the eye, the nose, and the mouth, right? And what science has discovered that is that there is an enzyme in human body that accepts SARS-CoV-2, a receptor. Yeah, a protein receptor called angiotensin converting enzyme 2. We call it ACE2, angiotensin converting enzyme 2. That ACE2 is much in our eye, our nose, and our mouth. So that ACE2 must interact with another protein on the SARS-CoV-2 virus called spike protein. Are you getting my point? So when the two of them come together, the ACE on the human body will be the one to accept SARS-CoV-2 spike. And once it accepts it, it allows it to get into our body, where SARS-CoV-2 can produce many of its own copy. So now you are to design a drug that will stop the entry of SARS-CoV-2 into the human body. Therefore, your target is either you target ACE2 or the spike protein, right? So it is advisable for you as individual to have an understanding of the structure of the spike protein and the structure of the ACE protein. Do you get the point? So we are still going to do that practically, but I'm just trying to explain. So because the drug you want to make must be complementary in shape to either the spike or the ACE before you can stop the two of them from interacting. Am I making sense? Hope my point are clear to you guys. Okay.
Yes.
So to do that, you can, okay, let's say we have 10 drugs drug A, B, C, D, E, F, 10 drugs. Let's say 1 to 10 drugs. You can get your SARS-CoV-2 protein from vendors, right? You can go to the lab and start testing drug 1, drug 2, drug 3, drug 4, drug 5, up to drug 10. And maybe at the end of it, if you are lucky, maybe 1 or 2 may be working against, may inhibit the proteins, right? And in many cases, none of the 10 drugs will probably work. And at the end of it, you discover that you just waste your time and you waste your resources. But thanks to AI, that thing that you're going to do in the wet lab, you can try it out on your laptop, on your computer, using some software, some algorithm to predict, okay, is the shape of this drug 1 to 10, is it complementary to the shape of the active site of ACE2 and spike? Do you understand my point? So computer will tell you this thing that you are bringing, the shape is not complementary, and when they bind, it's not going to have effect on the protein, or it's not going to have effect on ACE2 or spike. Can you see how computer? So before you even go to the lab, you already know which of the 10 drugs is going to work or not. So instead of you trying 1 to 10 drugs in the lab, computer will tell you, okay, from this I'm saying, possibly just 1 or 2 of them will work. So instead of you wasting your whole time working on 1 to 10 drugs, you only focus your study on the 2 or the 3 or whatever number that has been predicted by the computer for your experiment. I believe you guys understand my point now. So drug design frequently but not necessarily relies on computer modeling techniques. Yes, it frequently relies on computer modeling techniques. This type of modeling is sometimes referred to as computer-aided drug design. Yes, when you use computer to model what is going to happen in the lab, you model it on your laptop, that is called computer-aided drug design. So you can look at the structure of a protein, especially the active site of the protein, and say, but what I'm saying, this amino acid, a drug with this shape will probably work. And people have done that, and they've done that to predict a lot of even vaccines and drugs in the market. Now, so computer-aided drug design represents a computational method and resources that are used to facilitate the design and discovery of new therapeutic solutions. That is it, right? So drug design with the help of computer may be used at any of the following drug discovery. So let me just now, sorry, 1 second. I want to today. I know many of you may be saying, Dr. Dewi is talking too much. Yes, I agree. But my talk is going to be for today, just trying to lay foundation and for you to understand what I'm trying to do. So drug discovery may require any of these steps, right? Molecular dynamic simulation, batch screening, structural activity relationship, right? Molecular docking, molecular modeling, quantitative social activity relationship, pharmacophore modeling. Most of these things, we are going to touch them during the practical session starting from next week, right? So this I won't go into detail of this, but by the time we start, you will understand all this in detail, right? So now, this is something I want. We have drug design can be in 2 forms, on 2 standards. Yes, let me say on 2 standards, right? Now, ligand-based drug design. 2, structure-based drug design. Thank you. Now, if you remember initially, I said the design of drugs, either using computer or you are working in the lab, it has to do with you having understanding of the biological targets. What disease are you designing drugs against? Is it against SARS-CoV-2, against HIV, against hepatitis, against whatever? You understand? That's that. Now, there are 2 approaches you can apply. The first one is ligand-based. Ligand-based. In a simple term, what ligand-based has to do is you are designing a drug based on the knowledge of an existing drug. Do you get that? You are designing a new drug based on the knowledge of an existing drug. Let me give an example. If you want to develop a drug for curing fever and pain, right? You can look at the structure of Tylenol or ibuprofen, right? So let's say, for instance, let's say this is the structure of Tylenol. I'm just giving that as an example, right? You can see a benzene ring here, a benzene ring here, a benzene ring here, right? And you can see some other functions. You can say, okay, if 3 benzene rings are here, what if I design my own drug to have additional benzene ring, making 4 benzene ring, right? And some other functional groups. Is it going to work? Maybe the benzene ring is the one responsible for Tylenol curing headache and fever. So if this one has 3 benzene ring, if I should make my own 4, is it not going to work better than Tylenol or ibuprofen? That's your curiosity, right? And that is science. So if benzene ring is attributed to the action of Tylenol, you can say, okay, let me go and design my own drug too. But instead of 3 benzene ring, I'm going to make mine to be 4. So you are designing a new fever drug or anti-fever drug or anti-pain drug based on the knowledge of the structure of an existing Tylenol or ibuprofen. Do we understand that point? So in that case, there are different ways you can do that. There are machine learning, artificial intelligence softwares, right? That instead of you thinking, you can just put this Tylenol that you are looking at here. Tylenol, you will just upload the structure of Tylenol into that software, and it will give you a lot of compounds that have similar structure to Tylenol. Are you getting my point now? Am I straightforward? Can you before we go, any question from anybody? Maybe what I've been saying is you are not clear. Please ask question, please. I always like students to ask me question. Hello.
I have no questions.
Are you saying this to be interesting?
Yes.
So please ask me questions. So this ligand-based drug discovery is you are designing a new drug based on the knowledge of an existing drug. And that is why I give you Tylenol, for instance, right? Do you know during COVID, there are drugs that they are using for COVID because they use an existing drug from HIV, right, to design a drug for COVID. Ask me how. Thank you. Let me give an enzyme in COVID is the protease enzyme. The shape of the protease enzyme of HIV is the same. The shape of that enzyme in HIV in COVID is the same as the protease enzyme. The shape is similar, not 100% the same, but it is similar to the shape of HIV protease enzyme. So science now says, okay, if the shape of protease enzyme in COVID is similar to the shape of protease enzyme in HIV, then can we try a drug that is already known, FDA approved drug for HIV protease, and let's try it on SARS-CoV-2 protease. And that is why you see that what if you go online and see drug approved for treatment of SARS-CoV-2 of COVID-19, I'm telling you, you will see HIV drugs. Because logically, they believe if there's similarity in the structures of the two diseases, then the drug that is already being used to treat HIV, we can try it also in COVID and see if it's going to work. So what they also do is they now take some drugs from HIV like lopinavir, darunavir, nevirnavir, all those HIV drugs. They now go and put them on this machine learning artificial intelligence server, did more drugs with similar structures. And that is how we get a lot of drugs now. So when you like this example I give to you, if you have your own drug and it has 4 benzene ring, right? There is tendency that your drug that you are going to get will be more potent than already existing Tylenol. There is tendency. either similar or more potent than Tylenol, because you are designing your new drug based on the information of the existing drug. So when you put Tylenol into this server, it can predict hundreds of compounds with similar structure to Tylenol, and those new compounds are referred to as pharmacophore because they are structurally related. Structurally related to what? To that Tylenol that you are starting with. So that is how I can describe ligand-based drug design. Now, this other one, the other approach. So these are the two approaches for drug design anywhere in the world, ligand or structure-based. The other base is called structure-based drug design. So the first one, ligand-based, you design your drug based on the information on the existing drug, right? Now, this one, you are designing your drug based on knowledge of the structure of the biological targets that you want to work on. What I mean, when you want to design a drug against SARS-CoV-2 spike protein, you have to have understanding of how does SARS-CoV-2 spike protein look like before you can design drug towards it. That's structure-based. You rely on the knowledge of the structure of the biological target that you are targeting. For instance, if you want to design drug on RDRP, RNA-dependent RNA polymerase enzyme of SARS-CoV-2, you have to, on average, what is the shape of that protein before you can design your drug. So if you look at this diagram I have here, in this case, they are looking at this protein, MMA1 protein. I think it is one more or less like a cancer protein, right? Once they look at the structure of this protein, they were able to design, they are also able to do what is called virtual screening. Virtual screening means they do like molecular docking to see which of this drug is going to bind well with this protein. So, but in ligand base, you don't need protein before you design your product, your drug. You design your drug based on existing drug. That's ligand base. This other one based on this protein target. That's when you design. So if you look at this, this is a virtual screen like 100 of compound, and they dock it to the protein, and at the end of it, only one of it, called lead molecule, is the one that is giving them activity that the computer predict to likely work. And once they do that, they synthesize and they bring it to the lab. Right? There are that I'm currently doing in the lab right now that I have to use information on proteins of SARS-CoV-2s to predict my compounds. Now I've synthesized them. The next thing I'm doing now is to take them to the lab. Yeah, this is TB screening. Yeah, TB. This one is TB. So they see that this protein, when you inhibit or activate it, you can cure TB. Now, based on the structure of this protein, they are looking for different compounds that can bind well with this protein. When it binds well, it can inhibit. So when they get the protein, they take it to the synthesize. And when they synthesize, then the next thing is to take it to the lab and see if it's going to work. So for this drug, they say it has an MIC of 100 microgram per liter. So if this is inhibiting well, the next thing for them is to apply to FDA, do some study, more study, do animal study, do human study, and the next thing you see this drug in market. That's the process of drug discovery. But we're using computer-based algorithm, right? Any question before we proceed? Do you have any question? Please let me know.
No questions from my side, Dr.
I have a question.
Okay.
Okay, so as far as ligand-based and structure-based drug designs, are there
Sorry,
any
I can't hear you. Your voice is breaking.
Huh?
Yeah, speak now. You can talk now.
Okay, can you hear me?
I can hear you clearly now.
Okay, so I was saying, right, regarding to ligand-based and structure-based drug designs, are there any like particular limitations pertaining to those approaches? And if so, like how are they able to be addressed?
Yeah, there's there is no limitation to the two of them. Yeah, we are just using different approach. You understand, to arrive at the lead drug. So the only limitation, is can be for the ligand, for the structure base, because not all human proteins, not all disease proteins that we already have their structures. Yes, thank you for that question. You are right. So because this ligand structure base, yeah, it has to be based on the knowledge of of the biological target. And I'm telling you, not all proteins in the human that we know their structures. So if the disease you are targeting, there's no information about the protein or the structure of the protein, there's going to be some issue, right? So the limitation can be in two forms. One, you may know the protein, you understand? You have the amino acid, you have the sequence of the protein, but no structure of the protein. You have the protein, you have the amino sequence, but there is no structure of the protein. So that's one limitation. And how you can solve that, you can also use AI to solve that. There are AI and softwares, right? That when you copy the amino acid sequence of a protein and you drop it in them, it will give you a predicted structure of that protein. So when you have your target, you have the sequence, but you don't have the structure. You can copy that sequence to the software, and the software will model. It's called homology modeling or molecular modeling. It will model that sequence and give you the possible structure of that protein. So that's one challenge. The other limitation is you know your protein, you understand, but you don't have the amino sequence. You don't have the structure. So when you have that challenge, the only thing you can do is you yourself to start your own experiment in identifying the sequence and the structure, or you wait for other people to work on the sequence and the structure. If you don't have that, for that disease, you can't proceed. The only thing you can do is to go back to ligand-based approach for that disease. Do I answer your question, right?
Yes, thank you.
You're welcome. That's a very good question. That's a very I think there is this. I've worked on a protein on falciparum protein. That protein is not on protein data bank. Protein data bank is where we get the protein structures, and it's not there. So the next thing what we do is, okay, since we have the sequence, let's model the sequence ourselves, and that was what we did. But if we don't have the sequence and no structure, it's either we stop the project there or we start a new project to identify the sequence and the structure of that protein. Any other question, please? Any other question? Okay, so let's proceed. Okay, in fact, I even have it here. If an experimental structure of a target is not available, it may be possible to create a homology model of the target based on the experimental structure of the related structure. Can you see? So if the structure of the target is not known, it is possible to create an homology model of that target based on the experimental structure of related protein. So when you have the sequence of that target, of that protein, and you feed it into this AI software, based on the amino acid sequence that the AI is looking at, it will look for another proteins that have similar sequence, similar amino acid sequence. And it's going to use that to create a structure for that protein that you are looking at. Yeah. So, and that is referred to as homology modeling. Homology modeling. Homology modeling, also known as comparative modeling of protein, refers to constructing an atomic resolution model of the target and an experimental dimensional structure of related homology protein. So we are going to do that by the time we start our project. So okay, let's just continue. So as we've explained about computer-aided drug design, some of the advantages is this: time it makes our time to be effective. Imagine you have, for instance, there's a project I'm doing right now. I work on over, I think, close to 1 million compounds. I'm trying to work on a target, and I want to see out of this 1 million compounds from a database, how many of them is going to cure this, is going to work against this disease. So imagine I'm doing my experiment on 1 million compounds. I just take compound 1 to the lab, it's not working, compound 2, and I'm going to 1 million. Imagine the time it's going to take me. So what I did was to use computer-aided drug design, right? Do structure-based design. And out of this 1 million, do you know, I only have 20 that I am going to work with in the lab next week. So imagine the time it's going to take me for 20 and the time it will take me for 1 million compounds. So it makes our time, it makes our time to be efficient. So instead of taking too much time, AI with computer discovery, we make it to have a shorter time to do your work. Number 2, cost. Imagine for me to buy a kit for the experiment I'm doing, 1 kit is going to be for 5 compounds, is going to be like close to $1,000 for 5 compounds. Now imagine, so if I'm working on 20 compounds, that means I'm within like 4,000, 5,000 for 20 compounds. Imagine I'm doing that for 1 million compounds. So cost effective is the advantage of using computer drug design in drug discovery. What about accuracy? Yes, it also gives you accuracy. It also gives us information about the disease. Yes. For instance, you have the sequence, you don't have the structure. It can give you information about it, right? Screening is reduced. It reduces your screening time. It makes your time to work on database, and less manpower is required. So those are the advantages of using computer drug discovery in medicine, in science. So don't forget, this computer drug discovery we're talking about, they are the work of artificial intelligence that has been transformed into computer-aided drug design. So even different fields now use AI. Even people in architecture, they apply these in their structural design. That is that. Now, I list some drugs that has been discovered using this concept in the past. So all these drugs, they've used AI to discover all these drugs, and there are many more. I'm telling you, they use AI to discover all these drugs that you are seeing. AI was used to discover them. Captopril in 1981, saquinava is HIV drugs, dorzolamide, indinava, ritonavir, they are HIV, thiofiban, zanamivir, oseltamivir, retaguinavir is also an HIV for integrase. All these, they use AI to design them. And I'm telling you today, right now, either you are in Johnson & Johnson, you are in Merck, they have all those big pharma companies, they have a big section, a big laboratory section for computer-aided drug design and for structural biology. In fact, I can tell you there is no big pharmaceutical industry that does not have a section for computational or AI-based drug discovery section. There is none. There is none. So if you're a pharma student and you are taking this direction, it's a good point. It's a good one for you. So these are also useful links that you may need, right, to get this done. So these are also useful links so that you can also for additional reading, right? Do we have any question? We need to pick up the question so far, maybe on the AI that we've spoken about in the past, or on computer drug discovery. Maybe anything is not clear, please. I want you to feel free to talk to me. I'm just using today to lay, this is our first class, right? To lay this foundation for us. Then next week, so we are going to see the practical of it. So let me just say this. This project is going to be in 2 forms. One, I want to introduce us to Python programming, right? Secondly, I want us to work on computer-aided drug discovery, right? So I've not decided which one should come first. I will decide that between now and our next class. So maybe we should just start with Python, how you can learn a Python script, writing it yourself, doing it yourself, or we should start with the AI. So I'm going to decide on before our next meeting, which is next week Friday, on which one to go for. Right. So do you have any question, please? Any question from anybody? Any question? Does that mean everything is clear? Okay, I think we have a question on the chat. Okay, someone said no, sir. Okay, okay. Any question? Another question I want to ask you is this. Another question I want us to deliberate on this. Let me stop sharing, stop recording.